All right, so I think I'm ready to get started. Good afternoon, everyone. So my name is Gabriel Sawakuchi, and I'm from the Andes. I'm actually from Brazil, but I've been living outside of Brazil for a long time. I'm a physicist by training, but I'm a biologist by heart. And today I'm going to talk about one of the projects that we're doing in the lab. That's how to modulate metabolism through radiosensitized lung cancers. So just very quickly disclosures. And first, before I started, I just want to acknowledge a lot of the collaborators that helped with this research, and mainly Dr. Bright and Rishab. So these two people, they were the one that actually did most of the work here. So then, before I just want to introduce to this audience radiation therapy. So radiotherapy can come in very different forms, including different types of radiation. And these different types of radiation, they can have very profound consequences and different consequences as well. In terms of the physics of these two different types of radiation, I'm illustrating here a curve of the dose as a function of depth. And here you can think depth in the body. So here is at the entrance of the body, and at some depth is at the tumor. Photon radiation, that's here labeled 6 MeV X-rays, these are the conventional type of radiation that we use for most of the radiation therapy treatments. And you can see that the dose has a peak and then decreases as a function of depth, but never stops. So it can go across the tumor and still deposit those downstream of the tumor. Protons, now recently in the last 20-25 years, has been increasing the use, mainly in North America, but also in Europe and Asia. So there's about now 150 or so proton therapy centers worldwide. And just at MD Anderson, where I am from, we have 2 proton centers now. So protons, what happens is that the dose at the entrance is low, and then increases and has a peak, and after that peak has a very sharp fall off. So this peak, we can change the energy of the proton beam to modulate where this peak stops, and we can make it stop right inside of the tumor, independently of the depth, because we can modulate that. But the good physical characteristic of this is that it stops. So you can spare all critical structures or normal tissue that is beyond the proton beam. So with that, you can, just as examples here, produce much more confined dose distributions that you can spare much more healthy tissue. So this is a comparison of a pancreas treatment using photons and protons. So you can see that there's much less dose that goes beyond the beam for protons compared to X-rays. And this is another example for brain treatment. So then, I'm talking about this because this is very important when we want to combine radiation with any systemic agent or any drug that is delivered, administered systemically. Because the drug will also affect normal cells that's in healthy tissue. So we want to spare that as much as we can. So then protons seems to be a better way if you want to combine with some drugs that are administrating systemically, because if you're going to sensitize normal tissue as well, if you're, you can avoid this additional toxicity with protons. So then I'm going to also define another physical quantity here that will be very important to understand the biological mechanisms that I'm going to be talking about. So dose is the amount of energy deposited per unit of mass. But there's another quantity that characterizes the different types of radiation, that is the ionization density, which is defined as the linear energy transfer. So the ionization density, or the LET, describes the spatial pattern of energy deposition. So how the energy is deposited locally. And for photons, that's the conventional type of radiation, the ionization density is pretty much constant as a function of depth. On the other hand, for protons, at the entrance, the ionization density is small, or the LET is small, And then as a function of that, increases and increases until it reaches a maximum. And what's important here is that there's a differential in LET, a differential in ionization density, where at death or at the tumor, the ionization density is high, and in normal tissue, the ionization density is low. So low LET is defined, or low ionization density, for radiation that has an LET that's less than 2 keV per micron. So keep these units in mind as well, because sometimes I'm going to be referring to that. So, and this will be all clinical photon beams and high energy protons, that will be protons at the entrance or protons at normal tissue. While high LET will be protons at the tumor volume or other types of particles that are also used for cancer treatment, which includes carbon ions, alpha particles as well. So now to put this into the context of the size of a cell, this is a micrograph of a cell, where you can see the cell nucleus here. And these are radiation tracks. In this one here that's high LET, you can see that the track has these dots that, the dots are the energy deposition, where they are very compact, so it's very close to each other, so the ionization density is really high. While for low LET radiation, the ionization density is very sparsely. So you have an event of energy deposition here, and then the other one doesn't happen until here, and another one here, and so on. So it's very scattered how the energy is deposited. So then what are the biological consequences of this? Now, if you put in even smaller scale and look at our main target, that will be DNA for cell kill, what happens is that for low LET radiation, the probability of causing a direct DNA damage is much lower than for high ionization density or high LET. But all types of radiation, they induce all sorts of cellular damage that include DNA damage, damage to the lipids, and damage to all subcellular structures as well, including mitochondria and so on. So as I mentioned, low LET, they're sparsely ionizing, and what happens with low LET radiation is that there is oxygen or water molecules inside of the cell. And the low LET radiation will interact with these molecules and produce free radicals or reactive oxygen species that will then, in turn, interact with DNA and damage DNA. So then for low LET radiation, usually the tumor response is dependent on the oxygen status of the tumor. So if a hypoxic tumor is more resistant to radiation, the main reason is because of lack of oxygen to generate this reactive oxygen species that would in turn interact with DNA and damage DNA and then kill the cell. On the other hand, high LET radiation, because of its ionization density, can cause direct damage to the DNA. And therefore, high LET radiation is less dependent on the oxygen status of the tumor. But the other important aspect of high LET radiation is that it produces very complex and clustered lesions that are much, much harder to repair than low LET radiation. So the hypothesis in my lab is that to combine these physical aspects of radiation with biological mechanisms. And with low and high LET radiation, what we can do, we can manipulate the radiation beam such that you produce different types of damage in normal tissue versus the tumor. So in the tumor, more complex, hard to repair damage that can induce a very specific pathway for DNA repair. That's different from normal tissue. So then when you combine with some systemic agent that's targeting the repair of the type of damage that's happening in the tumor, that will be less toxic in normal tissue. So then, as I mentioned, radiation induces transient ROS. So while radiation is on, it's generating free radicals, so ionizing molecules, water molecules, and oxygen molecules, and producing these reactive oxygen species that can indirectly damage the cells. Radiation may also modulate ROS through induction of mitochondrial dysfunction. So mitochondria can be dysfunctional by direct damage by radiation, but also via radiation damaging DNA that then the cells need to repair, and the cell needs to produce nucleotides and energy that will also modulate the metabolism of the cell. So higher LET radiation also produce more ROS, and high LET radiation have a higher relative contribution of direct versus indirect damage than low LET radiation. So now, changing gears and talking more a little bit of the rationale for why we were doing this in the lab. So lung cancer is the most common diagnosed type of cancer worldwide. Also is the #1 in mortality, and 85% of lung cancers are non-small cell lung cancer. The 3-year survival improved from 21% in 2004 to 31% in 2017, and this improvement was in part attributed to advances in screening, diagnosis, surgery techniques, and target therapy, therapies that exploit tumor-specific vulnerabilities. So how non-small cell lung cancer are treated? So radiation is used in about 50% of the cases, either for cure or palliation. There is some inoperable early stage, that small tumors, and those small tumors can receive ablative doses of radiation. That's the conventional treatment. But also for the inoperable locally advanced, usually chemoradiation is used. And for fit patients who responded to chemo-RT, also immunotherapy, and usually is anti-PD-L1. For metastatic patients, it's chemotherapy and palliative radiation therapy, and recently target therapies and also immunotherapies. So targetable alterations with molecular target therapies that are approved, at least in the US by the FDA, involves several targets. But the only ones that has a significant amount of patients with those targets is KRAS and EGFR, in which about 25% of lung cancer patients, they have mutations for KRAS, and between 19 and 30% for EGFR. All the other targets in which drugs are available, the percentage of patients that are very, very small. So in radiotherapy, we don't use any of this molecular information that we know to either prescribe radiation or to combine with drugs. And how we prescribe radiation is only based on the stage of the disease, the performance status, and comorbidities of the patient. So patients that are not selected based on tumor genomic vulnerabilities, and these are not used to indicate synergies between radiotherapy and molecular target drugs. So compared to medical oncology, radiotherapy field in terms of using biology to actually improve the treatment is very primitive. We give the same dose for any patient independent of the type of the tumor or mutations in the tumor. So we become interested on this KEAP1 gene because this KEAP1 is mutated in about 15 to 30% of patients. So this is TCGA data to show that there's a very large incidence of this mutation or alteration in this gene for lung cancer. But also, And interestingly, patients with this mutation, they usually do poor compared to having wild type, this gene wild type. And this is for overall survival for all types of treatments that these patients receive, including radiotherapy. And we also look at cell lines from the CCLE database, and also notice that for non-small lung cancer cells, in that data set, it's over 30% of the cells were mutated for KEAP1. So recently, a group from Stanford, they published this data on patients that received radiation, and conventional radiation, that's CRT here, or high-dose radiation, that the SABR here, where patients that have mutations in KEAP1, they also did worse than patients that didn't have that alteration in terms of local recurrence, but also overall survival. So they have also like a control group in which these patients, they only underwent surgery, and there was no difference between the status of KEAP1, which indicates that KEAP1 actually make the tumor radio-resistant. So let me talk a little bit about the mitochondria now, but very briefly, because I think this audience is very aware of this. So the mitochondria is the end effector of the cells and synthesizes many of important biomolecules that's very important for radiation response, including nucleotides, fatty acid, amino acids. Also converts oxygen into energy and produces reactive oxygen species, in which in the right balance is good. It helps with important cellular signals. And mitochondria has ways to keep the right balance of ROS by producing ROS neutralizers. So now some background on KEAP1. So KEAP1 modulates metabolites that neutralize ROS. And KEAP1 binds to and mediates the degradation of NRF2, preventing its translocation to the nucleus. So if KEAP1 reacts with ROS, it releases and stabilizes NRF2, which then translocates to the nucleus, promoting the transcription of several antioxidant genes and glutathione. So glutathione is one of the main neutralizers of ROS. So KEAP1 mutation results in active NRF2, and in this scenario, cells continuously utilize glutamate for antioxidant production and supplying the TCA cycle with substrates. So KEAP1 mutated cells heavily rely on glutamine through glutaminase 1 to maintain adequate glutamate levels. So glutaminase 1 is at the center of glutamine metabolism to produce glutathione, but also alpha-ketoglutarate that feeds to the TCA cycle to manufacture nucleotides. And glutamine is the most abundant amino acid in the body and is a substrate for many biological molecules. And as I said, GLS1 is at the center of glutamine metabolism. So disruption of GLS1 in KEAP1 mutated cells disrupt glutamate supply for antioxidant production, which leads to excessive ROS and results in DNA damage, lipid peroxidation, and ferroptosis. And I'll talk a little bit later about what ferroptosis is, but it's a form of cell death. Disrupts glutamate supply to the TCA cycle as well, which can influence production of molecules that are important for radiation response, such as ATP, lipids, and nucleotides. So cells that are KEAP1 mutated, they are addicted to glutamine. So if we disrupt glutamine metabolism in those cells, then we see drastic consequences. So GLS1 catalyzes the conversion of glutamine to glutamate. And end products of GLS1 include glutathione, that, as I mentioned, is a reactive oxygen species scavenger, and nucleotides as well. So in my institution, MD Anderson, we have a drug discovery platform, and there we do things from discovering the drug to launch clinical trials with these drugs. So we discovered, or this platform discovered GLS1 inhibitor. That is what I will be using in most of the rest of the presentation here. So this GLS1 inhibitor now is in phase 2 clinical trials. We already tested toxicity, and there's only minimal toxicities that are manageable. And there were some efficacy as well in a phase 1 clinical trial, so we're moving for phase 2. But the goal is also to try to combine this GLS1 inhibitor with radiation. So the rationale for this talk is that lung cancer has relatively high KEAP1 mutation rate. So these tumors, they are relying on glutamine turnover via GLS1. And patients with KEAP1 mutated tumors do poorly when treated with radiation. So perhaps KEAP1 mutated tumors can better cope with radiation-induced damage through larger capacity of neutralizing radiation-induced ROS. So the question is, can disruption of glutamine metabolism radiosensitize these KEAP1 mutated cancer cells? So that's what we're attempting to answer. So some of the data here, so these are several cell lines, all or most of them, they are non-small cell lung cancer cell lines. So the ones highlighted here in the box, they are all KEAP1 mutated. And you can see that they're sensitive to the drug as a monotherapy. On the other hand, cell line that's not KEAP1 mutated, KEAP1 wild type, it's responsive to the GLS1 inhibitor. Those are mouse cell lines, and we don't know the KEAP1 status of them, but some of them show some sensitivity, some of them don't. And this is a normal cell line that shows moderate sensitivity to the GLS1 inhibitor. In terms of radiosensitization, these figures are cholinergic cell survival. So here is the survival fraction as a function of radiation dose. And the curve in black is not treated with the inhibitor, it's only radiation alone. And the curve in blue is radiation plus the GLS1 inhibitor, where you can see sensitization. So this was for photons, and the plot in the middle is for protons. It's the same cell survival curve for the same cell line, but instead of exposing to photons, we expose them to protons. The figures on the bottom, they're the same sort of plots, but a different cell line. Okay, so it's also a KEAP1 mutated cell line. And this plot here is just quantifying the survival curve to measure the radiosensitivity. And what you can see here is that the inhibitor, the GLS1 inhibitor, sensitizes the cells to both photons and protons. And you can see there here as well, that's a phenomenon of protons that we discussed this before, is that the ionization densities of protons is higher, so it produces more complex damage and has a higher efficacy to kill cells. So just protons alone compared to photons alone, for the same dose, you produce a higher kill effect. And when you add the drug, you radiosensitize even further. And you can see the same trend for the other cell line as well. So here's just quantifying how much sensitization it was generated by adding the drug. And you can see that was sensitized by 1.2 to photons and about 1.5 for protons. And the different cell lines also shows the same trend. And what's interesting is that the way protons interact with this inhibitor is different from the way that photons interact with this inhibitor, because the relative sensitization for protons is higher, and this is what we're measuring. So the relative sensitization of the inhibitor caused by photons versus protons, where for some reason there is a higher synergy when we combine this inhibitor with proton beams compared to photon beams. And then to demonstrate that indeed KEAP1 radiosensitize or make cells radioresistant, we obtain a cell line, a cancer cell, lung cancer cell line that was KEAP1 wild type and knock KEAP1 out. And you can see that the cells became more radio-resistant. And this is the case for photons, and this is the case for protons as well. And then when we treat with the GLS1 inhibitor, we can more or less recover the radiosensitization. And again, this is the same sort of plots I showed before, just to characterize these cell survival curves to show that we were able to sensitize. But in this case, for this cell line, we didn't see any difference on the effectiveness of protons sensitizing more than X-rays. So for this cell line, the proton synergy was not different than the photon synergy. So then to try to understand the mechanisms of this, first we try to understand what's this GLS1 inhibitor is doing in terms of glutathione production. So we measure the glutathione levels in the cells. So the lower the ratio of reduced to oxidized glutathione, the less the cells can respond to oxidative stress. So as expected, GLS1 inhibition disrupts glutathione homeostasis. And this would lead to lower tolerance to oxidative stress. So this is the case for the two cell lines that are KEAP1, cancer cell lines that are KEAP1 mutated. For the cell line, this is a normal cell line. We didn't see an increase or a decrease in the ratio of GSH to GSSG, and it seems that reaches a plateau. So it's less sensitive to the GLS1 inhibitor. So then we also looked at oxidative stress, where we measure total ROS produced in the cells, but also only mitochondrial ROS. So here, this is a bunch of data. I'm going to try to navigate you guys through this. So these are different treatment types where we didn't treat the cells with anything, and at different time points, 24, 48, and 72 hours after radiation. Then we treated with the GLS1 inhibitor or radiation GLS1 inhibitor. So here is photons with the GLS1 inhibitor and protons with the GLS1 inhibitor or alone. And we looked at total ROS or mitochondrial ROS. And we saw that total and mitochondrial ROS increase after GLS1 inhibition and also after radiation. And the increase was higher for protons. Also, this increase persists for up to 3 days after radiation. And the effect could be partially rescued after treating with a raw scavenger, that's NSA here. So this then is related to oxidative stress, but what we're trying to understand now is what's the mechanism of that. Then we also look at lipid peroxidation and ferroptosis. And so ferroptosis is a cell death mechanism that's induced by lipid peroxidation. And lipid peroxidation increases after GLS1 inhibition and radiation. And also the increase is higher for protons. And again, the increase persists for up to 3 days after radiation. And inhibition of ferroptosis partially rescue the survival after treatment with the GLS1 inhibitor and in photons. So we cannot rescue the effect completely, and that was the same case for the raw scavenger, but we can partially rescue the effect. We also look at alpha-ketoglutarate supplementation, because alpha-ketoglutarate is also at the center of glutamine metabolism, because it feeds to the TCA cycle that would then generate all the important things that is needed for DNA repair. And what we found is that alpha-ketoglutarate could rescue the survival of the cells completely after treatment with the GLS1 inhibitor. And we think that this, because alpha-ketoglutarate feeds into the TCA cycle and leads to the synthesis of the molecules that are important for radiation response. So that's what we were trying to understand now. What's the actual role of alpha-ketoglutarate on this process? So then we also measure DNA damage or double-strand break damage by using gamma-H-ray-X. And here, again, are different treatments and different time points from radiation. So 4 hours, 24 hours, and 72 hours after radiation. And we see that for the cells that we treated with radiation and the GLS1 inhibitor, in general, we have a trend of higher double-strand break foci. And this persists even for a very long time after radiation, up to 3 days. So what we think that the mechanism related to this is so first, have these cells that they are KEAP1 mutated. So they have a higher capacity of dealing or coping with ROS. So they usually have a higher background of glutathione on them. Then you expose these cells to radiation. So while radiation is on, is causing DNA damage, the cells are trying to cope with the DNA damage by repairing. So the cells will have an accelerated metabolism, which in turn will also generate more reactive oxygen species. And then once the radiation is off, the cells still maintain a much higher level of reactive oxygen species because of mitochondrial dysfunction that I mentioned before. And mitochondrial dysfunction could be related to coping with DNA damage by repairing the DNA, but also could be directly, mitochondria could be directly damaged by radiation as well. So then in this scenario, if we use a GLS1 inhibitor that would then block the production of glutathione, which is the mechanism for the cells to protect themselves from damage caused by the reactive oxygen species that are being produced, then we can sensitize the cells. So to show that that was the case, what we did an experiment in which we treated the cells before or after radiation with the GLS1 inhibitor. So we treat the cells 6 hours before radiation with the GLS1 inhibitor, or 2 hours or at radiation, but we also irradiate the cells and then we treat later on with the GLS1 inhibitor. And we show that indeed there is also an effect when we treat the cells after the fact, which indicates that the mechanism is indeed related to this higher level of ROS that these cells are now producing because of radiation damage that's making the mitochondrial dysfunction. So then we can apply this inhibitor even after radiation. We can still get a reduced sensitization effect. So then to show that this is the case also in vivo, we use a cell line. In this case, the cell line is not keep on mutated. And we did an experiment with mice where we exposed to radiation alone, or to the inhibitor alone, or the combination between both. And we saw a moderate effect when we combine radiation plus the inhibitor compared to radiation alone. But you know, we don't see a high effect because remember, this model is not keep on mutated. We're working on developing the knockout of the cell line to be able to do the experiments properly now. But we see that even for a non-mutated keep on wild type cell, this inhibitor also reduces sensitizes. And here is to demonstrate that glutamine 1 inhibition is important, but glutamine metabolism is what is the important aspect to reduce sensitize. So in this experiment, what we did was to inhibit a glutamine transporter, and we were able to sensitize cells that are keep on mutated, significantly sensitize the cells. But also in vivo model, we also were able to sensitize, radiosensitize this tumor quite a lot. So then the take-home message is that KEAP1 mutation makes cancer cells more dependent on glutamine metabolism. KEAP1 mutation makes cancer cells more radio-resistant, and this is in part due to their larger capacity to cope with oxidative stress. Also, disruption of glutamine metabolism with GLS1 inhibitor radiosensitize KEAP1 mutant cancer cells, and proton radiation have larger synergy with GLS1 inhibition in KEAP1 mutated cancer cells than photons. And this radiosensitization appears to be in part driven by oxidative stress through amplified DNA damage and lipid peroxidation as well. And as you can see, there's a combination of different mechanisms that is actually radiosensitizing the cells. The radiosensitization also appears to be also heavily dependent on alpha-ketoglutarate. And the mechanism of radiosensitization may be due to radiation-induced mitochondrial dysfunction, which is amplified when we use a GLS1 inhibitor. So non-small cell lung cancer patients with KEAP1 mutation, they have poor outcomes, and no target therapy is available for this cohort of patients. So then this cohort of patients may benefit from combining radiation with a GLS1 inhibitor. So the next step of this work is to produce more robust data to support a clinical trial to combine this GLS1 inhibitor that was discovered at MD Anderson with radiation in KEAP1 mutated patients. So I'd like to acknowledge all the members of my lab. So they were the ones that did all the hands-on work on this, and also my collaborators that contributed to the work as well, and the funding agents for supporting this project and also other projects in my lab. So thank you very much, and if you have any questions, happy to try to answer.